Here, the researchers talked about the two different types of adaptive cell transfer approaches. First one is T Cell Receptor based approach, and second one is this Chimeric Antigen Receptor approach. This is more commonly used nowadays, that is Chimeric Antigen Receptors, because it has better functions than this TCR or the traditional approach. So we'll discuss about these two kind of approaches. So idea is that to take the T cells from the blood and then transduce them with either this T cell receptor based lentivirus or chimeric antigen receptor based lentivirus. It can use one can use other viral also. And the difference in the T cell receptor is that the TCR is basically expressed on the activated T cells naturally, and it has this alpha and beta chain that helps in the recognition of the tumor cells antigen. One of the mandatory requirement in this T cell receptor based approach is that the antigen information need to be presented by the antigen presenting cells to these T cell receptors, and then only these T cells will respond to those antigens which are present on the tumor cells. So in this case, one of the main mandatory requirement is that antigen processed and presented by this major histocompatibility complex. And once these T cells are primed by the tumor antigens that are present in the patient, then these T cells will be expanded and then reinfused into the patient after this chemotherapy. And we have understood the reason for the chemotherapy many times in our previous videos. So this is how this T cell receptor based approach works, or their mechanisms is like that. And now we will quickly or briefly go over this chimeric antigen receptor based principle. So in this case, the mandatory requirement of the APC is, or the antigen presenting cells, is not required. Here, direct tumor based antigen receptors are presented on the T cells, and these chimeric antigen receptors will be linked to the intracellular signaling in a way that will activate the T cells. So intracellular signalings can be like only the activations of T cells using CD3 zeta, or it can also have the co-stimulatory domain. That is, the two co-stimulatory domain that commonly used are this 4-1BB or CD28. So they can be used either, chimeric antigen receptors can be used with either of the one co-stimulatory domain, or both the co-stimulatory domain can also be used. And this will make these CARs as different generations. First generation use only the activations intracellular signaling, that is CD3 zeta. The second generation used one of the chimeric and one of the co-stimulatory domain. It could be either 4-1BB or CD28. And the third generation used both the co-stimulatory domain along with this intracellular signaling. In the second generation also intracellular signaling is there, but there is only one co-stimulatory domain. There are more generations of the CAR has also been developed, which include more signalings along with this co-stimulatory domain and activation signalings to activate the T cells more aggressively. So idea is to activate the T cells in a way that it will act only on the tumor cells aggressively, but it will have a minimum or no impact on the healthy cells. So once these T cells are being transduced with this chimeric antigen receptors, they will also be expanded and then reinfused into the patient after the patients are preconditioned with chemotherapy. So this is the two difference between TCR and ACT. There are some facts that has been observed on the preclinical trial about the T cells, that naive T cells has a better in vivo persistence compared to the mature or activated T cells, and this has been found in the preclinical trial results of mouse and monkey. And this could be the reason people preferred naive T cells for this T cell receptor based approach or the chimeric antigen receptors based approach. And one more thing that they have found out is that the CD8 plus T cells and CD8 plus T cells can directly kill the or directly trigger the apoptotic in the target cells by releasing of the cytokines. And the one fact that people have found out, which is paradoxical to the CD8 plus T cells function, is that its ability to release the cytokines is inversely proportional to its anti-tumor efficacy. That is, more cytokines it will release, the less anti-tumor efficacies it will have for the future reactions or for the future interactions with the tumor cells. And the reason that they said that there's more its ability to proliferate, its ability to release cytokines will accelerate the differentiation process of the CD8 plus T cells into a different lineage. So CD8 plus T cells has also been termed in this paper as like a stem cell state of a T cells, which has a ability to proliferate and differentiate it into the different populations. So this is what the authors has presented in the paper about the difference between T cell receptors and the chimeric antigen receptors, and some of the observations that they have seen in the preclinical studies. That's all for this video. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. Thank you for your time.